Hello everyone, welcome to our video on brain dissection. I'm Dr. Michael.
I'm Alina.
I'm Stefan.
I'm Tantan.
So we're only going to go through the details which are relevant for the 3420, the Anatomy and Physiology lab course. And I'll be talking about removing the meningeal layers of the brain.
I'll talk about the fissures and lobes.
I'll talk about the inferior view.
And I'll talk about the sagittal cut.
And I'll talk about the various coronal cuts. I hope you enjoy our video. So the purpose of this video is to show you how to take off the meninges covering the sheep brain, and also to remove the periorbital fat which is found here. And by doing that, you'll be able to appreciate the various fissures on the cerebral surface and the sulci as well. So we're going to start by trying to identify the optic nerve. So this is an optic nerve. You see right here, it's an optic nerve, and so I'm going to cut the fat which is surrounding it, and that's the periorbital fat. So and so you see I've removed all the fat surrounding this optic nerve. I'm going to do the same for the other side as well. Right, so we see the two optic nerves, and notice I'm cutting off the remaining periorbital fat. So here we have the two optic nerves. Now what you see here is the meningeal layer, and we're going to remove it by cutting starting here. So I start at the cerebellum, and notice that right here is the pituitary gland, and I'm going to cut around it because we want to keep it intact while we examine the brain. And so I just cut around it. And if you notice, this part of the meningeal layer is going to be especially tough, and I'm going to cut a little deeper there. And whatever I did on this side, I'll repeat on the other side as well. Okay, now you can just peel off this entire meningeal layer, and you see it just comes off like a cap. There's one more spot. Right, there we are. So we have removed the meninges. This is the dura mater, and right below that would be the arachnoid mater, but you really can't see it clearly because in this preservative it would have dissolved. And what you're seeing here is the pia mater, which is intimately associated with the cerebral hemispheres.
This part of the video aims to help you identify the various fissures and sulci of the brain. From that, we want to help you identify the lobes of the brain and their associated functions. First off, we have the longitudinal fissure running anterior to the posterior aspect of the brain, and it separates the right and left cerebral hemispheres. And then we have the transverse fissure, which separates the cerebrum from the cerebellum, and it runs here. In the anterior aspect of the brain, we have the central sulcus that separates the frontal from the parietal lobe, and it runs laterally across both hemispheres. Directly anterior to the central sulcus is the precentral gyrus, which is where you would find the primary motor cortex. Directly posterior to the central sulcus is the postcentral gyrus, which houses the primary somatosensory cortex. The parietal occipital sulcus would separate the parietal from the occipital lobe, but it's not readily apparent on a sheep brain, but it would be found between the division of the occipital lobe, which is found here, and the parietal lobe here, so it would run laterally across both hemispheres again. And this is the occipital lobe here, which is where you would find the primary visual cortex. Here we have the lateral sulcus that separates the temporal lobe from the frontal and parietal lobes. And deep to this, you can find the insula, where you would find the primary gustatory cortex. And in the temporal lobe, you have the primary auditory cortex.
This part of the video aims to show you the structures of the brain that are visible from the inferior aspect. So first we have this pair of olfactory bulbs, and this gives rise to cranial nerve 1, which is responsible for the sense of smell. Then we have this pair of optic nerves, and so this is going to cross over at this X right here, the optic chiasm, and then this continues on into these optic tracts.
Visual information will travel through the optic tracts and eventually end up in the occipital lobe.
Now we have this midbrain. Now usually the midbrain will have this pituitary gland that's going to cover it and prevent you from seeing it, but we removed it from this brain. So here we have this midbrain, and if we follow the midbrain, you can see these 4 bumps. So these top 2 bumps are the superior colliculi, and these bottom bumps are the inferior colliculi. The superior colliculi play a role in processing visual stimulus, and the inferior colliculi play a role in processing auditory stimulus.
The midbrain is the most proximal part of the brainstem.
As we continue down, we see that this first bump right here is the pons, and then this oblong shape inferiorly is the medulla oblongata. And then as we continue, we see the spinal cord.
This part of the video aims to show you the structures of the brain that are visible from the mid-sagittal cut. To perform this cut, you want to cut along the longitudinal fissure of the brain, and when you open it up, you get the sagittal cut. So if you look at the brain from the mid-sagittal cut, first you can see the thalamus, which is the circular structure right in the center of the brain. Inferior to that, you can find the hypothalamus, which gives rise to the infundibulum and then the pituitary gland. Posterior to the thalamus, you have the pineal gland, which is shaped like a P, and this is part of the epithalamus. The pineal gland plays a crucial role in the regulation of sleep-wake cycles. Posterior to the pineal gland, you have the midbrain.
Inferior to that will be the pons, which is the bump right here, and then inferior to that you can find the medulla oblongata, which is the oblong shape right here.
Superior to the brainstem, you can find the cerebellum, which has very clear gray and white matter, and the white matter are branched sort of like a tree, so that's why it's called the arbor vitae, which means the tree of life. Next, we'll talk about the ventricles of the brain. Here, you can very clearly see the lateral ventricles, and inside the lateral ventricles, you can find the choroid plexus, which are the specialized cells that make cerebrospinal fluids. The cerebrospinal fluids flow from the lateral ventricles into the third ventricles, which you would find in this area, but you can't see it from this cut because the sagittal cut cuts through the wall of the third ventricles. So it will be here, and you can see that when we do the coronal cuts. From the third ventricles, the cerebrospinal fluids flow into the cerebral aqueduct, which is the ridge you can see right here. From there, it flows into the fourth ventricle, which is the space between the cerebellum and the brainstem. And then here you can also find more choroid plexus. From the fourth ventricle, the cerebrospinal fluids flows into the central canal,
which is part of the spinal cord.
This part of the video aims to show you the structures of the brain that are visible from the coronal cuts. What you can see first is the gray matter, which is superficial, and the deep white matter. And you notice that this structure here is the corpus callosum, and it is connecting the two cerebral hemispheres. And as you see here, you see the two lateral ventricles, which are inferior to the corpus callosum. And if you notice, there is this very thin membrane which is separating the two lateral ventricles, and that is the septum pellucidum. The septum pellucidum would be visible from the sagittal cuts as well sometimes, but it's very clear in the coronal cuts. Now, if you were to go more posterior, you will see that you see the corpus callosum here, and inferior to the corpus callosum is the two lateral ventricles, and here you can even see the choroid plexus present in these lateral ventricles. Now what is inferior to the lateral ventricles is the fornix right here. This is the fornix. And here you notice the thalamus. And when you go even more posterior along the coronal cut, you see that the corpus callosum is still visible. Here are the two lateral ventricles, and you notice there are these two seahorse shaped structures, and that is the hippocampus, and that plays a role in membrane consolidation, and that's what you're going to need in order to pass this exam. And if you look here, you see there's the thalamus, and right here in the middle you see the third ventricle. And now it makes sense why when you did a sagittal cut you could not visualize the third ventricle.